Hello, my name is Dr. Roderick L. Roll, and today I will be talking about molecular basis of inheritance. The key to replication is the complementary structure of the two strands. Remember, DNA is composed of two strands. It is properly referred to as a DNA molecule. So a DNA molecule has two DNA strands that run in opposite directions, and we call that antiparallel. It is held together by these hydrogen bonds. So the key to the replication is the complementary structure. How does replication occur? Well, Messelson and Stahl addressed this question. They proposed three hypotheses to address it. The first hypothesis was that conservative replication took place. Then the second one was that dispersive replication took place. And then the third was semi-conservative replication. This shows the depiction of the three models. The first one is conservative replication. In this case, the old strands stay together and two new strands that are light blue are going to be replicated from the old strand. With semi-conservative replication, one old strand is paired with one new strand. And then in dispersive replication, it appears that the fragment, the DNA is going to fragment into portions, and the portion that is missing is going to be filled in with new DNA strands. This is Messelson and Stahl's experiment. Step 1. E. coli was exposed to heavy nitrogen in the growth media. This means that every E. coli that replicated would have to use nitrogen in order to make its nitrogenous base. Step 2, nitrogenous bases contain only heavy nitrogen. So every E. coli that was replicated would have the heavy nitrogen that is N15 incorporated into the bases and incorporated into the adenine, incorporated into the thymine, the cytosine, and the guanine. So all of those nitrogen bases had heavy nitrogen. In step 3, E. coli was removed from the heavy nitrogen media and placed into another flask that had the light nitrogen. Light nitrogen had the N14. So as the E. coli replicated, it would now incorporate the light nitrogen into its genome. So E. coli was used because it replicates really fast. Within 20 minutes, you go from 1 E. coli to 2 E. coli. So originally it was in the heavy nitrogen flask. When you remove it, every nitrogen that is composed of the DNA would be N15. But now after you put it into one round of replication, the light nitrogen is now going to be incorporated into the DNA. So the results after Messelson and Stahl destroyed the bacteria, therefore releasing the DNA, was he saw that there was a medium band. If it incorporated only heavy nitrogen, you would have one heavy band in this test tube. If it only had N14, it would be a lighter band. But what their results indicated that they had a intermediate band that was in between N14 and N15. This meant that that round of replication created a DNA molecule that had one N14 strand and one N15 strand of DNA. Step 5. E. coli was then exposed to light nitrogen for a second round of growth. Step 6. Results indicated one light band and one medium band. The data from the experiment suggests that semi-conservative replication took place. One original strand will pair with one daughter newer strand. This was clearly observed after the first round of replication. So where is this DNA that we've been talking about? The DNA is located in the nucleus of a cell. DNA can also be located outside of the nucleus, inside of mitochondria, or if it's a plant, inside of chloroplast. One chromosome is also referred to as one DNA molecule. Now, when that chromosome replicates during interphase, now we're still going to call it one chromosome, but now it's going to be two DNA molecules. Each DNA molecule has two strands of DNA. This is the structure of a eukaryotic chromosome. It is composed of 8 proteins that are called histones. Collectively, it makes up another protein complex called a nucleosome. The DNA is wrapped around that. DNA can be supercoiled into a structure that is called heterochromatin. This is typically seen during DNA replication, versus the DNA can exist as euchromatin. Most of the time it's in the true state, euchromatin state. This is when protein synthesis can take place. During replication, new strands of DNA are synthesized in the opposite direction. We refer to this as antiparallel direction. In the picture below, to the far left, that is a DNA molecule which is composed of 2 DNA strands. The 2 strands are going to be separated. What holds those 2 strands together are hydrogen bonds, a very weak bond, easily to break. Then once we have severed that connection, we can then put new strands in between. Those new strands will be complementary to the other strand. Each nucleotide strand serves as a template for building a new complementary strand. Notice that the old strand and the new strand move in opposite directions. That's what the numbers stand for, 5 prime versus 3 prime. The unreplicated chromosome is wrapped around nucleosomes containing 8 histones. Tightly packed heterochromatin becomes less tightly packed heterochromatin in order for replication to occur. It is tightly packed, but the enzymes have to be able to maneuver themselves in between these nucleosomes and histones. So now we're going to talk about replication on the molecular level. An enzyme called helicase is going to break those hydrogen bonds that holds the antiparallel DNA strands together. That is illustrated by this green blob that's moving along the zipper in this image. Primase will then synthesize the RNA primer on the 3' end. The third enzyme, called DNA polymerase III, is going to synthesize the leading strand. Now, when we talk about DNA polymerase, we have different types. In reference to bacteria, we're going to use DNA polymerase 1, 2, and 3. If we're talking about humans or eukaryotes, we will use DNA polymerase alpha, beta, gamma, delta, and epsilon. So to keep this simple, we're going to use bacteria replication. So the DNA polymerase 3 is going to synthesize a continuous leading strand. It will therefore start at the 3' end of the primer that was just placed there by primase. The leading strand, once it starts, it will continue until it is finished replicating the entire sequence. DNA polymerase 3 adds nucleotides to the 3' end of the primer. The primer is in red. DNA polymerase 3 is going to read the top strand in the direction that we call 3 to 5, and it is going to synthesize new nucleotides in the direction called 5 to 3. This helps us understand directionality. In reference to 3' and 5', they're talking about the position of the sugar. So if we look at the nucleotide, which is composed of a phosphate group, a deoxyribose sugar, and a nitrogen base, only look at the sugar. The sugar is that 5-sided structure called a pentagon or a pentose. We labeled it carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Notice that carbon 3 is facing the bottom of the screen, whereas carbon 5 is facing the top of the screen. If we rotate this 180 degrees, then the 5 would be facing towards the bottom of the screen, and the 3 would be facing the top of the screen. So that's all we're saying when we say one direction is 3 to 5 and the other direction is 5 to 3. This slide is looking at RNA. RNA is going to be composed of a different type of sugar than DNA. So the sugar is called ribose and not deoxyribose. At position carbon 2, instead of a hydrogen being in that location that I circled, we're going to have a hydroxyl group in that position. So that's the only difference in the structure of the sugar. So since we know that RNA is different, it has to be removed eventually. Now we're going to discuss the lagging strand. In this image, it would be depicted in the picture on the bottom. The lagging strand is going to be a discontinuous synthesis versus the top strand was continuous synthesis. Primase is going to come in and create primers on the lagging strand or on the template that is going to produce the lagging strand. DNA polymerase 3 is going to start at the 3' end of the primer and synthesize new nucleotides until it reaches the adjacent primer. Once it reaches that primer, it is going to stop. That is one Okazaki fragment. DNA polymerase 3 is going to then detach from the lagging strand. DNA polymerase 3 is then going to attach to the second primer. Then it will read from 3 to 5 and synthesize from 5 to 3, starting at the extreme 3' end of the primer, and it will make the second Okazaki fragment. Cycling will continue until the entire lagging strand is synthesized. DNA polymerase 1 is going to come in and now remove the primer, and it will replace the primer's nucleotides with DNA nucleotides. DNA polymerase 1 removes the primer in the 5 to 3' direction as it reads the template from the 3 to 5' direction. I labeled it 1, 2, 3 to show you which nucleotide is removed first. That blue nucleotide 1 is removed first, and it's removed from 5 to 3 as DNA polymerase reads the bottom strand from 3 to 5. DNA polymerase 1 replaces RNA with DNA while adding nucleotides to the 3' end of the adjacent nucleotide. DNA polymerase 1 continues until it reaches the last nucleotide of the primer. Then it will leave a free hanging 3' end gap. Another enzyme called DNA ligase will come in and seal the gap by joining the 3' end to the adjacent 5' end. During replication of DNA, we're going to create what is called a replication bubble. And in this bubble, we have two ends. On the left end, you'll have a replication fork, and on the right end, you'll have a replication fork. As helicase proceeds in my image from right to left, it's going to create a supercoiling effect. So the difference between the top image and the bottom image is you see the supercoiling effect. If that occurs, this consortium of enzymes are not going to be able to move. They're going to stop. So you have to somehow alleviate this supercoiling effect. Topoisomerase, which is also called gyrase, will alleviate the supercoiling by nicking one of the strands, and you will unravel the knots. Single-stranded binding protein will stabilize the unwound strands. This holds the fork open. Compare and contrast prokaryotic replication to eukaryotic replication. In prokaryotic replication, you have one bubble with two forks. The bubble is referred to as the origin of replication. In eukaryotes, you're going to have many bubbles or many origins of replication and many forks. This is bacterial replication. Notice that once you start at the origin of replication and you open the two strands and make a bubble, replication will take place going up and down at the same time. Therefore, replication will take place for that whole circular chromosome, but you only have one replication origin. Whereas eukaryotic chromosomes are linear, you'll have multiple replication bubbles.